fresh talent. What's that? Yeah, could go over there for parking lot.
All right, welcome to dive 587 of the remotely operated vehicle SuBastian. We just touched down on the bottom for this dive. We are at a site called Roca Redonda, and we are going to be making our way, and we are already just starting this dive expecting some interesting life here. So it's going to be a good dive. I'm actually going to dip out real quick and go grab dinner because also just started. So we're going to have Jimbo jumping in here, and I will be back momentarily. But welcome to dive 587.
Johnson. I know, I'm a lot there. Yeah. They're all shrimp. Are these shrimps? Yeah. I see a dandelion sapphire, cesta clams, and then these little. Whoa. Actually, he's almost in a great position right now. You can probably stop that. Just start pumping. Another dandelion. Hey, are we?
Can we zoom?
Yeah, we're gonna zoom now. Oh yeah, full zoom.
So that's Thoracica genus.
Can we have a zoom of the shrimp?
Copy that, ROV.
Whoa, we will. Whoa, we will. Throw in the trap. Okay.
No
I've not
way.
seen this before.
Yeah, there's a possibility of that happening.
That's what I was wondering. Yeah, there is a possibility of that. Hello.
Let's see what long known eyes.
So, she says between 6 and 6. So, and it looks like they're already making the order. 6 to 7. Yeah.
There's some gravity ones there. The top left has some. Oh, they have lots of eggs in them. And,
Where are they at?
they have to. They're orange colored on the end.
Yeah, you can see it. Then we just go around just look at yeah.
That's a really nice picture right there.
Hold on, yeah, that's excellent.
What?
Oh, finished by guy rolling.
You want to see? What you said?
Yeah, go for it.
Yeah. Thank you.
Yes. Yeah. Like rapid decision making.
So those flat traps, anemones are probably eating the shrimp. They get stuck in there. And there's these colonial siphonophores, those orange golf balls that like they like areas that are being decomposed and old
And they're
scavengers.
closed right now, so they're munching away.
They're probably eating some shrimp. Yeah, I'd be surprised.
We should tell Stuart to come and have a look at this. He's probably
It's amazing.
fine fish. Is it eating too much?
Fat fish. What's that coral?
Sorry, I shouldn't call it. Where in the stream moving?
Well, those are Caribbean shrimp. I don't know what further than that. I can't tell.
Shall we vacuum some?
Be fine with me. Have the lasers on, please, for a second? Yep. Okay.
The laser on.
They're on?
Yeah.
Sorry. I gotta find them.
Okay. There's one here.
Got one.
It would be great to collect some corals in this area. This is unique.
Okay, yeah. but we already decided that the area is.
Oh, yeah, but no.
Okay, we've got some prelud worms too.
Where's for biodiversity? Remember that we also doing biodiversity.
Okay.
Yeah. For the
But
northern rivers.
how are we doing time-wise and sample?
Well, we haven't started yet, so we can take a couple of samples.
Okay, okay. And I think we should factor some
I
of
think
these.
some of these should be great
Yes,
samples.
that's exactly what we're thinking with that.
Okay, great.
For the Darwin.
Yeah.
You know which species and the Antobacter there looks great.
So let's find a coral that we want to sample that looks good. I have to go.
Shh.
Yes. Alright.
Yeah.
Solitary hydroid.
Yeah, we're gonna take
A piece of that one.
a piece of the yellow and a piece of the white, right?
And shrimp that's there.
A piece of the big one. That's the difference.
Ceratopollo and the shrimp, that's different.
A piece of the big one.
That's a nice
Isn't
picture.
that amazing?
But it's getting lit up pretty good with the.
That's a,
It's
that's
like
an associate growth. Associate shrimp.
Yeah.
It's a lot of
Really?
different. One of the currents.
There's definitely current.
Yeah.
It's not as bad as before. Like, look at that. But it's not dragging the day too much. It's like current.
Is that? Siphonophore. T-Rex, hell yeah. Could be, yeah. T-Rex, good. Philopelia is what it's called. What's that? Oh, it's weird. Superior palates, we have a really high limits for male big shrimp, and the siphonophores that are pushing on the outside of the field.
Yeah.
It's just,
The only thing
I
we're
don't see,
missing is the bacterial plants.
yeah, I don't see the ultra brock
So
yet.
what about this kind of veneer?
Yeah, right.
Maybe that's what the shrimp are eating.
Yeah.
Maybe
yeah,
that is a bacterial plant, huh?
yeah.
Apparently someone just
Yeah, I was gonna ask if you.
What's that stringy bit over there?
That rock
It's
question.
kind of net,
Yeah.
spider net, I don't know.
Don't see the sulfide, don't see the alteration of the rock really. There's maybe some iron staining in it.
We would look for a yellow colour, right?
Well, if it was just elemental sulfur, that's what you see, like, kind of like that.
Yeah.
Yeah. It's true. But I would expect to see iron oxide.
Do you think that these should do some kind of dial migration? They're just kind of hanging out until they can pop off.
It's amazing, right?
Did you see this guy? Oh,
I don't know,
you see
these
the
are
other
pretty,
guy? Can you-
that tend to be benthic.
There was a different gastropod. Can we go up a tiny bit? Para arriba que había otro insectito.
See the gill there.
That guy, you see this
Ah,
guy?
yeah.
You see the white guy? Who's
Yeah.
this?
It's an amphipod, no?
The isopod? Yeah.
It's cute.
Alright, I'm out of here.
Those are great.
Do you guys want some more zoom on somewhere?
I mean,
Close
you've
ups.
you've done a nice job. I don't know, you wanna collect here?
Uh,
Is that what you're
I
doing?
Well,
think
the
we-
girls were saying about the Gorgonian type coral, perhaps the Anthomastus as well, and perhaps the socket of the shrimps.
Right.
Yeah, so we just
That's what I
grab
was
a
thinking.
couple of representative samples of what we see here.
Yeah.
So perhaps if we start with maybe the shrimp sucking,
Can we try
Yeah.
to use one shrimp, one, two
and then
shrimps?
the Anthomastus,
Start around the
and then move to the coral.
Okay.
Let's just go one shrimp. Good fall.
That dandelion, so those are the funny ones.
Yeah.
Each one of those little
Can we come on board?
arms, not the tentacles, but the things in the ball, are individual
Yeah.
organisms.
Mm-hmm.
When they come together, they
Do they
have some
move
different functions, right?
along the colony?
They move.
Yeah.
Some are built for swimming, some are built for digestion. They come together.
I think also it makes it easier to communicate.
Oh, I don't think so. I don't think so.
Okay, so
They're already inside the tube.
they're already in the tube. I'll see.
Should have just turned it on.
Yeah.
Yeah, just see how many you can.
Alright.
What's up?
Yeah, we're gonna zoom now.
Yeah. Tiny one.
This is jar 1.
Jar 1.
Yeah.
Okay.
They're swimming away.
Oh no, they're on the top. Okay.
They're quick little guys. Look at that. Come on, come on, come on. Yeah, you got a couple. Sure.
There's a couple of them.
Definitely got 1.
Yeah, we got, I think, 4.
See them.
That's not bad.
Come down a little bit, there's some big ones there, yeah. There you go. Try to get one with it.
Oh yeah, they're in the jar.
Yeah, yeah.
Okay.
Sample 1.
How many would you like?
I think that's
1
good.
kilo.
Thank you.
That's fine.
Breeding shrimp. Fish,
More?
This is gonna be
Event 1.
Okay.
one SS1.
Yeah.
Oh, the index works so good now. Alright, so that's jar 1.
Okay, perfect. Thank you very much.
Thank you.
And then
want to sample those
yeah, we'll take the Anthomastus as well, which is
Okay.
the red.
So you see the reds. If you can zoom out a bit.
Okay.
Show you that.
Oh, you want to suck it?
No, we can just take it with the palm.
You
And
might be able
it's
to pick up the rockets on it and see how large it was.
so.
Can
I'm just
you
gonna-
stuff this thing so I can put
Yeah, I'm
this
gonna let
on?
him just throw this here real quick.
Yeah.
What do you want, David? Maybe.
Alright.
Ah, right, it's there.
But like, new thing. Let's sit on that.
Well,
No, it just takes a little getting used to it. But yeah, like that. Alright.
Alright, so you're looking. No, go for it. Sorry.
Yeah, no, it's all yours.
yeah. Yeah, that thing here, right?
What do you want to take after us?
But then we're saying taking a representative sample of those corals here. So it's not for the RNA later, it's just a.
We know what it is. Yeah, that's cool.
Samples of commonly observed corals on the way.
For the red one?
Yeah.
Alright, I'll put this,
I think I have to move.
yeah. Let's see if I can.
Where's
There's a rock that I
Okay,
can
so
get.
we've got the RNA-lated corals,
Oh,
which
see
are
the
those
rock,
five,
yeah.
and
Up, up,
then
up
they
here.
want a sample of the biodiversity across the entire thing. So it's taking
Okay.
commonly observed corals.
This will find out where we're putting it first, because we have to put it in the cord show. Where do you want the sample?
This red thing's on there.
So, where would you?
How do you want to sample it?
Where do you want to put it?
We just launched one trip. Or lost power to this. Hold on.
Put it in a coral. Yeah.
right. Well, thanks to Jimbo for letting me grab dinner right as we touch down here.
See the white one?
For folks still trickling in, we're pretty early in this dive. We've been live for about 15 minutes. And as you can see, we touched right down and already a ton of life right here. This is dive 587.
Nice. Nice. So that's this really cool.
So what happened? My dog was shut down. My drugs turned off.
For this dive, we are kind of going to work our way up the slope of a little islet here. Roca Redonda, or the round rock. I can actually show you what it looks like if I can find it here on the camera.
And put it in where?
There we go. So that is, whoop, lost it there. That is the round rock. And so we're going to be working our way towards that, making our way kind of up the slope.
Well, that's the advanced side of sea, but
Starting pretty deep and just working our way up. So we're going to be from about 950 to 220, I think is about what we're shooting for. You can see quite a steep little structure there. So it should be good.
Not seeing any alteration of the
Okay, so then
One is that one or more rocks on the rock walls that can be a bit dangerous.
my thought was just, it's one of those ones that, you know, if you grab it, it's kind of sensitive, so I was like, if I can get it on a rock, would you prefer that if you don't care?
I mean, it's something that, forming the rock, so it's hard to fill out these rocks, but then you have to be careful when you put more rocks on the rocks, and by shape somewhere.
I also think that maybe changes impression with audio.
Yeah, that's fine, I just didn't want to,
We do a zoom on the
remember when we grabbed that one and grabbed it and messed it up?
Yeah.
Let's definitely have a, see how
So while we're getting ready for a sample here, I'll give you a little bit closer of a look. So we're kind of exploring, we're going to explore this little knob here. We're going to just kind of head up and over, and then just be working our way up slope there.
Yeah, it's just, I think if I let
Pretty
it go
steep.
fall in front, alright,
Should be a fun dive. Doing video transects along the way, as well as collecting coral and lava samples. There might be an intermission here or there. It'd certainly be a good area for one. But yeah, still early time for the dive here.
went down. Going down was problematic, but as long as it's fine.
It was the
Current?
tactic to this.
No.
Going down.
Okay.
It really changed it a little direction. Yeah. So did
Yeah. That's what Rob was saying as well, yeah. 180 degree swaps in the current.
So the cable was
Well, it looks like there's deep stuff that's interesting if we get too shallow and we can't.
yeah. The start of the dive is amazing.
Yeah.
We already have shrimp.
It's pretty cool to see all the shrimp habitat here. The one that's a commensal along the coral up here is a different species, but it's all so it's on the coral. Even with all this available whatever food, these guys are feeding on it.
Maybe these guys are just horrifically like.
Yeah, they are cannibalistic.
Yeah.
No, temperature?
I've only seen a couple of crabs.
I haven't seen any.
There's a lot in there.
maybe come straight down on it. Come over here. Did you get me?
There's that shrimp on the aftermath. Did you see that?
That you have it
Okay.
there, yeah.
See it?
Yeah, I see him clinging on.
It's the same as the others. You got video of a crab on top of a antibasis polyp eating out of the polyp. Stealing dinner from antibasis.
But the rocks are all loose.
Yeah, that maybe like it's kind of the reefs.
Goodness.
Could be a dead reef.
Debris fields. Yeah.
Good pick.
Oh yeah.
Awesome. Thank you. A shrimp went into the cube.
Yeah. We'll have some vagrants today.
All the effort to slurp one and then it just goes in.
It's
Oh no, it goes back
Oh,
up.
see you later, mate. Well, you've got your associate, which is the main thing.
They look like Alvin Karas.
There is one.
Yeah.
So you do think so?
Well, I can see from the gross morphology, it looks Caribbean. It looks like Alvin Karas, but that one certainly did.
But isn't it that they should have like this border?
That's just a certain genus. The Rimicaris
Ah, okay.
genus has that.
Oh no, your shrimp's gone.
Yeah, see this game.
Yeah. Oh no, he's
No,
gone.
no.
in there.
Yeah.
Ooh.
Nice.
Event 2.
Yeah.
Anything else? Anything else in the sample?
Yeah, the little bit of the coral to the left of it
You
as
see
well.
the yellow and
same-
white corals?
in the
They were
same-
together in the
Up left.
In a different coral
quiver?
quiver.
Yeah.
I figured you'd be in here.
You reckon? So
Wait. By Manjari.
I thought you'd coke. So I don't see any alteration on these rocks.
Is that?
You see the alteration on these rocks?
No,
oxidation or anything?
It's talus, but it could be manganese on it.
Yeah, oh, yeah,
there's
manganese.
Coral quiver 1.
So where do we go?
So we are on the first waypoint.
Uh-huh.
See this data here?
We started today. We
I
found
was
Basically, looking at these now.
So we diffuse the field that's here.
Okay.
What is
First waypoint. Temperature's fine. There's nothing much. Like
all of it?
rail layer on the same. So yeah,
Oh, yeah. Ah,
Where
okay.
you want to put this next one? There's two over
Are we
there.
already at waypoint
Yes.
1?
There's two
Ah,
curves.
yeah. So that we are just here, and we have to go across. Yeah.
And then we start with the vertical
Lot of exciting people
feature.
are here.
On top of it, and then we see what we can work on.
We want to try to go on this peak. There's a little peak right here.
Which is where the number 10 is, 110.
Well, it's just off that. So
Yeah,
we're
yeah.
gonna go across this. The
Across
peak is
that.
right in the middle.
Okay. Okay.
So when I
Grand.
was going to look for rocks.
You want the rock of this?
Not this.
Yeah,
I
on
wanna
the
see if
peak.
there's a real rock face.
Yeah, yeah. Because this is just all tumbled and
It's all talus.
Okay.
It's
Eight to 10.
other than that,
No, we were it was that. That's what I was going to do. I know.
So we touched down right as dinner time started, and we got a watch change. A lot of commotion right now. For folks just tuning in, welcome. You're just catching the beginning of dive 587.
Even when it's not active, it'll be active. These will come up. It will go quiescent.
It is a little toasty in here, isn't it?
Lots of people getting very excited about it.
All right, I'll come back around.
Yes, but you're aiming for like higher, right? Once it gets
More solid. Yes.
Yeah,
I
okay.
need to see actual
Yes.
rocks on the cliff rather than just tumbled rocks.
Okay, cool.
Yeah.
But we do want to sample that white fan
Yeah.
coral. Where is it
Yeah.
going to stick?
Yeah. So,
Thank you.
Could we get a good zoom on it before
Thank
we
you.
grab it?
So
Yeah, which quiver do you want?
which quiver? Not number 1. And not number 10 and 12.
About number 2.
Number 2?
Okay.
Yeah, we'll get a good zoom on that. Should be. Hello.
I'll be your assistant for a few minutes.
Yes, and Salome will be here soon to hopefully for some dinner. I love these fly trapping enemies.
you do.
Yeah. If I was a sea creature, I'd want to be a fly trapping enemy.
Mm.
Not a sponge.
The active skyfly, they're in the Gulf of Mexico and all this sediment, they get all dirty. And gross, but
They just
they are
look like
cool, though.
tanks of the sea, aren't they? Just
Yeah.
They did collect sediment, like, just nuts.
Yeah.
They look like one of those little gummy sticky hands from a quarter machine.
Exactly.
Dropped in the dirt. It's all I can ever think of
Nobody
when I
knows.
see them all covered in sediment.
Everybody knows I'm a deadly predator.
You know, so that density of anemones means that maybe they should have been here for quite a while, right?
Yeah.
So if we sampled a bunch of these shrimp, you can tell if there's been multiple recruitment events.
What's the temperature here?
I thought it was 5 something.
5.2?
Okay, let's keep an eye on that then.
5, yeah.
Should we do a quick snap zoom before we? It's up. It's up.
There we go.
Yeah.
Yeah.
On the
See
shrimp.
that shrimp?
What's the white thing
It's a
behind?
Bathy Palmanella.
Wow.
There's 2 of them. 3 of them, maybe 3. Oh, yes.
Those are really
It's so
happy.
rare to get them on the outside like that. Do you mind if I just have
Oh,
a
but
moment?
There's isopods in there as well.
Yes.
What did you say the genus was?
Oh, it's Bathy Palmanella.
Bathy Palmanella.
Palmanella. You see the big forearm?
Yeah.
Well, that's the
Oh,
characteristic area.
it's like he weightlifts on one side only.
Just on one side.
Yeah.
Oh, that's
Like
right.
it's got a big palm.
Yeah.
I've got a nice picture of that.
There's one top left too, a bigger one.
That's a very strange looking coral, isn't
Yeah.
it? Maybe we can snap the bit with the shrimp on, hey?
So is the larger appendage, is that not a sexual dimorphism? Both have it, or is this like a?
It can be in the group. The ones I've seen are male.
No, okay.
Yeah.
I wasn't sure if they aged out or not.
No. Not that I know of.
Yeah, if possible. Oh yeah, it goes. Yeah.
Before we do a sample, we zoom in and we take an observation. We can also use that
Because they see the polyps too.
so I'll do another one here.
Oh,
Click it.
I think that's the maximum marking dog.
Some of the colder water we've been in.
Yeah, it is.
And then,
It's also some
yeah,
of
it
the
takes a
deepest,
snap
no?
photo, all the metadata,
Yeah.
everything in one go. Then I can just
Curious
say
when we
like,
did that thermocline on the way up.
That's a nice shot.
yeah, I did. For,
Yeah, great. Thank you so much.
Submit.
You
That
are
creates
kidding.
a new
The
line.
currents around here are no
you
joke.
can create a login quickly.
Ooh, what's the thing at the top?
Okay, great. Yeah, so then you can actually
yeah.
click on these pictures and they all.
What?
Oh, it looks like it might be a tinaphor.
Wow.
It
On the
got
top?
caught,
Oh,
got
yeah.
caught in the coral.
Oh yeah. Something.
It's like a tinaphor, Medusa or something.
There's like a tiny little ghost shrimp on the top as well, no? Is that something on the here?
Yeah, I'm staring at it.
Yep, I see.
You see something right there? Yeah, it's
This guy, yeah. Decorator prawn? Yes.
We're just about to sample this, and this will be event 3.
Event
So we'll
3.
let you to it.
Okay. What role am I doing? Am I doing your role?
Yeah, because
Yeah, thanks for holding
Kara's
down.
learning.
Sorry for being late.
It's okay.
Nice
Enjoy.
job.
I was like slipping my soup like, okay.
Thanks for letting me on here.
Sorry? Is there a preferred piece you guys want, or just a...
I don't think the shrimp will stay on. So if there is a piece with any associates
Yeah.
with these isopods, the other little, they were tiny crustaceans.
Yeah.
There's one white
White,
isopod
yeah.
on the rock.
No, on the
Oh, you meant,
How you
if
doing?
you know,
You want a hand?
the shrimping things will tend to stay if you don't
Yeah,
move it
I think
quickly.
this, they're doing event 3
Okay.
now,
Very
right?
slowly and steady.
Perfect.
Did Amy say what this is going into?
I think there were 2.
Okay, perfect. So that shrimp is not the same kind of shrimp as the rocks.
See, that's the.
It's not the same as the one on the rocks.
It's
No,
not
it's
the
not.
same
It's
one.
not the same.
That's the beauty.
Ah, yeah, I see.
They're so specific.
No, I didn't spell that out.
Sometimes they fly off and they come back.
Coral is in a bit of a difficult plane for seeing what you're doing.
Yeah. Transects better to come top down.
So when are you on? Or are you on right now?
On 8 to 10 and
8 to 10.
midnight to 2.
Oh, yeah, we are getting a. Would you call that an account? You just put white optical. Ever down. Just color and class. Yeah.
Trying to pick which one to get.
Did they?
Or the one back.
Do you know what the yellow coral is?
It's probably a swift, yeah.
Those things are
Okay. From my area of the world, I only know red and white.
Okay. Yeah.
But
We
my
have
area
we have like
is not
we have a yellow contavertex here and yellow swifties here.
Okay.
That big of a piece.
Is that a picnic on it then?
This is where is Wally, right? Where do you see that?
This is
A picnic on it.
Where did you see that?
Bottom left, next to the yellow core.
Ah.
It looks like it. Yes.
Could be.
And like this. Yeah. Rotate.
Okay, I misestimated the size of this coral. I misestimated the size of this coral. I thought it was huge. And I've seen the coral. It's not that huge.
I have to adjust as you go.
Alex, upstairs in the kitchen on the screens, we cannot see the depths or anything like that. Is there a way of doing that?
I can put it on the front one.
This seems to be a tough one.
Yep. Just got it. Yep. I see it now. Cool.
don't.
keep the shrimp for you
I
guys,
don't.
but just have none of it.
Yeah.
I can now.
If I would be the
But usually
shrimp, I
what
would
we do
also
is we don't.
swim away.
Yeah.
We don't
Hang around.
fully click until we see it go into the pool.
Alright. Oh,
Because sometimes it might fall out, and then we have an observation, and we have to go and like figure out and like error.
Yes, I can.
So we just like wait until we see it going.
Temperature is fluctuating a lot.
Pardon me?
The temperature is fluctuating quite a bit. Whereas
What-
like just
what
in
is
the
it?
5 and now it's 5.17, that's not a massive change. But
Yep.
yeah, it's given that we haven't moved.
Yeah, and we're pretty deep.
Yeah.
Yeah.
Just do we have the CTD profile today?
It's here.
Does it match with what we see for the moment?
Have a look.
Yeah, it does.
It's just come up on the edge there, just a little bit. So just needs a little bit of stuff hanging on from that.
Is it a black hole?
No. No, this is some kind of, I think it's in maybe like the gorget or primrose.
Then I would go for it first.
I have a feeling it might be some kind of Maurice, Paramorice.
Right. Nice and down. Just put the cap on, we'll just push it.
So your latitude is 0.286769 and your longitude is -91.65024 and we are 931.6 meters. Oh, now he wants to come in, but too late. You've been on this boat before? You've been on this boat before?
This is my first time. It's three. I have a couple of cruises. So I think I'm going to this is one of the cruises.
So what do that? Everybody wants you on their boat.
It's really cool. It's like,
There's no one in the milk at once. You're like, why? Why? Yes. Gotta get the pieces and stuff. You can't be in Latin America. Nice one, Tyler.
Thank you.
Very nice. Thank you.
Did we already sample shrimp?
Yes.
Okay, brilliant. Oh, yeah.
What's next?
If you don't mind, if I look around a little bit
Sure,
to get a feel for the
absolutely.
place. I just saw one feature that I'm curious what it is. And then I think after that we will start traveling slowly towards the next waypoint.
And you know all that all from that?
Encrusting sponge? Ah no, that's the leftover of the Antomastus that people sampled.
And she's your student, right? Yeah. You supervise. Okay. And she's what unit?
Pretty strong corals, no?
Yeah.
Yeah.
And you're in Calvin as well? No, what the heck is
Oh, there's a tulip sponge.
tulip sponge?
Yeah, next to the primnoid.
Oh, okay, yes. I call that a lollipop sponge.
Ah, okay, yeah.
But tulip is a nicer one.
Oh. Well, when we open up the first one, like put something in, shrimp, so a lot of it go out. Just jump right in. Curious to
Yes. No, square is about this, right? Okay.
Are we moving on?
Yes, I think so. So you're happy to go in video transect mode? We can.
There's something definitely interesting going on here because usually I mean 900, it's a lot of diversity for 900.
Yeah.
Yeah.
I mean, I'm curious to see like the extent of the
Shrimp.
shrimps. Yeah.
Yeah.
Just not sure if we should be looking down.
Ooh.
Ah.
What are those? What are those massive corals there?
Yeah. Yeah, we can go deeper. We don't have to look at it. So there's no
Although
1,000 meter restriction. but-
Can we just have a look at those massive corals?
I don't know if they're corals, but yeah.
Are they huge anemones?
They look like.
Holy moly! You remember when I said that everything's massive around here?
Yeah, I just
They
remember.
can't move.
No.
Can we just have the lasers
No,
on
they
them?
don't.
The lasers are on.
Yeah.
They're just on the rock
Oh,
below.
okay. All right.
So,
So,
this anemone is about.
it's like 2 foot. Yeah.
It's about 30 centimeters.
Yeah. It's a big
That
one.
was amazing.
One's got its hair straight, the other's got the curly. Wow.
Wow.
Where is the clownfish? Come on. Nice. For a minute, I thought that was a black coral from a distance, but
Yeah, at first as well, when
Yeah.
it came up in the dark
Cool.
here, I thought it was a coral.
oh god, I forgot to take a biological shot. Hang on, let me just take.
Do
a
you
shot.
want me to zoom in again?
It's okay. I've got it.
Yeah.
All right, fine. I'll do it. I do another one. Got it. Thank
Cool.
you.
Thank you. That's nicely studied as well.
Actinarium.
Okay, then now we go on a video transect. We'll zoom in a tiny bit so that the poor student who has to count all the shrimp can actually count. And so why did you say they, no, you said there is lots of massive specimens here, and why did you say that might be?
Uranium. No, I'm kidding. I don't know. Well, I think the venting. I think the venting creates. We suspect that the shallow venting has something to do with it, given that that's the variable that makes this site different to all the other sites, and maybe it just creates some productivity that feeds everybody and makes everybody bigger.
Mm. Temperature has dropped again.
Yeah.
Yeah.
Oh,
From 5.2 something back to 4.98. Oh, Chrysogorgon?
Yeah. And stylostrate. Oh, that weird
Siphonophore.
jelly. Yeah.
Yeah.
What are they called? What's the general word for them? Those.
The little ball.
Yeah.
Yeah, so it's a dandelion.
Dandelion. That's right. Dandelion.
Yeah, they're real cool.
Dan says that usually when they are also associated with kind of venting kind of places.
Really?
Oh, there's another one.
Yeah. Because we saw them the last dive as well, next to Santa Cruz.
Yeah, true.
Yeah, Tempest back up. Sure, it's fine. No, it's bouncing around.
Black coral?
Yeah.
So just gotta jam it, put it down in
The
there.
one on the
You want to see the one on the
Do you want
right?
to, can you zoom out a little bit?
You wouldn't. Sorry, we're moving over. You want to
Okay.
zoom out or in?
Oh, out a little bit. Just. Oh, but now we're good. I can see everything. Okay, so that is that one is Aliasonos, Aliosinani, I think. Oh god, and these are two black corals.
And you know them.
Alciosonina, that's it.
I was more looking at the black coral.
Ah, yeah, the alcynation we have already.
I mean, we have. This is not the we're outside of the designated
Sampling zone, yeah.
sampling zone for OMZ. And in terms of diversity, I think we have that one.
We
already.
have that one? Okay,
Yeah.
we have that one.
Alright. Then we go again? Yep. Thank you.
But this is I would call this as a sparsely populated coral garden.
Or densely populated shrimp.
Yes, a densely populated shrimp field. It's a shrimp extravaganza, that's for sure.
What I love is they maintain like a distance. They're pretty evenly spread.
Yeah.
Yeah,
Like
that's
everyone's got their own little space.
Canadian style shrimp. No offense. Always gotta say that term.
They're all like nicely evenly spread
Yeah,
out.
yeah.
Some sort of competition avoidance.
yeah, I'm starting to think a point pattern.
Yeah, it's exactly how it's regular. Indicative of.
That's cool.
Do you want to look around and zoom in on things, or do you want straight video transects?
I think we can have a look around, but like if we pause the vehicle while we zoom, in case we don't lose ground.
Yeah, it just means.
Oh, yeah, knowing that we ground.
Ooh.
Might zoom out a tiny bit.
Evening, Chris.
Oh, wrong American. This is Tyler.
Oh, damn. Sorry. Yeah, new waypoint?
Yeah, new waypoint. We're doing kind of a slow video transect, so I'll probably just it'll be a bunch of small moves maybe.
Okay, no worries.
Thanks, Jason.
The subtle shade. I got a parasite there.
Another one of those, yeah.
Yeah.
Or the same one, but
I think it's a different one.
Yeah, we've passed quite a few.
So from the shape of the fans, it looks like the current, as you can see at the vortex, is really going across. Before we had
Definitely looks like that before, yeah. You can see it waving that way too.
yeah. Oh, is that Madrepura?
We got Madrepura, yeah.
Wow, that's deep.
I know.
Oh, another of these enormous anemones.
Yeah, mira esta anemone enorme.
Sí, había una.
Oh, look at this beautiful sponge.
You're just trying.
Sorry, yeah.
no, it's totally fun.
Wow, it's gorgeous. No, esa es la hora UPS con la que estamos operando todos. Ya lo noté, pero ya le. Eso es un primero. Wow, these anemones are mental.
They're amazing.
Crazy. ¿Cuáles son estos?
Si los que no tienen aleta no los dan en la parte inferior. O puede ser aleta.
La la. Sí.
Pero es que no tienen la parte.
Mira que se viene. Esta es como una cámara de más distancia. So we're also seeing the bottle brush.
Yeah.
Black corals, which I usually associate with like 300-meter depth, but there's the same food, I guess, that there is at this depth. What is that, shallow depths? Could that be why we're seeing these guys here? Oh, what's that? That looks different.
The yellow one, huh?
Okay, I think that it's, can we get a zoom on the large polyps? I think those are the ones that look like they've got Christmas lights on them. Is that a bad description?
Or the white one in the middle?
The white one in the middle?
Yeah.
Ah, no, I see what you mean.
Yeah.
This one?
Yeah.
The
I think, I mean, that's just a bit different of what we've seen earlier. I think that might be a clavularia growing on, it's a parasitic.
It looks like a sponge with zoanthids on it. No?
No, they are, oh my god, the name is escaping me. It's a type of coral that is, oh yeah, that's right, it's a sponge covered in ¿te acuerdas cuando me contaste que es de que no dije el otro día?
Ay, no.
Oh god, the name is escaping right now, but anyway, I'll take a picture.
Or anemones?
No, they are a type of Cnidarian that
Uh-huh.
are colonial and they grow on top of things parasitically.
Yeah.
The name is escaping me
There's
right now.
another one. Just I'll zoom out a little bit.
Wait, thank you.
And then there's another one just up there. On the top of my screen, which I think is the same kind of thing.
Ese
Yeah.
no esponja con...
Super.
It's getting to me now.
This is real expert flying. Pretty cool sponge though.
Yeah.
Zooming around thing.
And the yellow coral we have?
I mean, we
we-
have it on
we-
on a different dive.
Yeah.
On a different dive, but we could collect it.
That yellow? Oh, there's just
We
so
have-
many different yellow corals.
We have some.
We have that
We have
one.
that yellow one already, right?
The thing I was looking at was limpets.
I saw limpet, yeah.
Oh,
Yeah.
Do you know what these
Yeah.
guys are called again? The ones that
Yeah,
are growing
there's a
on top
couple
of them.
lipids.
The subcolumn?
Yeah, that's growing on top of the sponge. It's not Stolonifera, it's not Clavularia, it's another.
Got another dandelion teenophore under there.
Oh, yeah.
So there's two, but they're the same species, right? The one just has a
That
pollen
one there,
and the other one doesn't.
Yeah. Yeah.
And there is a kind of orangey one in the middle.
I know, we definitely sampled that one the other day. That was the one that
Okay.
I was like, that's new.
Yeah,
Okay,
but I think
Tim's.
that's something else.
This one?
Yeah.
Or this one?
Well, which one are you pointing at?
That one or that one?
They look the same, just
Yeah, they do look
different
the
colors.
same. That's what I was
That and
thinking.
that, yeah. That's what I'm saying.
But
Limpets.
the limpets like
Where are the limpets?
on the up here on
Oh.
this. Yeah.
This thing here?
So
Yeah.
Oh, okay.
when I was looking at peripheral vent fauna in Taiwan, limpets was one of the signals. And the Nucella like mollusks as well.
I mean, we can sample or move on.
Sorry, I just see something.
Yeah.
All the polyps closed. I leave the decision on sampling on you guys. If you need it, then you take it. If you don't, we move on.
So,
Do you think that's a Jamaica? Is that where you're going with that? Like a soft coral?
No,
we
we do.
So, from my understanding, we want biodiversity. It's always good. We're feeling like we have the time.
Mm-hmm.
Do we have the time?
We do.
All right, let's grab a yellow clip.
You want a clip of the bottom one or the top one?
Whichever is easier for them.
Okay.
It's the same
So
one.
if we can sample it, please.
Which one?
The yellow one.
The yellow one? Yeah.
I mean,
To me, well, I don't.
you feel like we have it?
No, I felt like we didn't have it.
Yeah.
I felt like the branching pattern was different.
I mean,
than-
even if we do have it, it might still be interesting given that we're so far away from deeper and we're so far away from, we're like in a total different bioregion.
But I've only seen half of what we've sampled.
Me too.
Yeah. Maybe we
Well,
haven't
maybe
seen
I've
the same
seen
half.
maybe I've seen it a little bit more because I
Processing them.
Yeah, I process. Okay, maybe I've seen 50% more. Maybe I have seen all of them. I just can't remember them.
They look so different when they're in the lab, though.
Yeah,
It's all the
they do.
yellowness. but-
Especially the yellow bubble.
Alright, yeah,
You guys done
we didn't.
the calls?
We're doing some more.
On that white
Oh, you're doing
cross.
some
We
more.
haven't made it very far yet.
What a surprise.
Yes.
I'll come back.
Yeah.
Thanks.
Shall I ping your message when I think it
Sure.
starts to get interesting?
Yeah, I'll be.
You'll
So
keep
I'll
an eye
look
on
at
it.
I'll look at this.
Yeah.
How many more calls do you think to say?
How long is it?
It's
in
not
the stream?
No. I think
mean-
we need to be. Well, otherwise we're never going to make it up to the top.
But I understood that it's fine to look around
Yeah,
and
that's fine.
to explore. Yeah.
No, that's fine. Yeah. I mean, we've never been to this depth in
Yeah.
this place, so that's.
Yeah.
Are we happy with core flipper number 3?
I think so. Yes.
Alright.
Lots of different ones to keep that.
Yeah, no, it's very different. Are you gonna tell us which rock you want?
Yeah,
You've already taken one?
No, I don't want this. It's chaos. I don't know where it came from. I mean, I know it came from someplace along here. I'm hoping that we'll come to a scar and get some samples of the section. If we keep going up and we only see tables, then I'm gonna have to make a decision.
Yeah. Okay, sorry, Belen wants the white one, the big white one.
Okay.
The big white one. You mean
Yeah,
the
the
Bermuda?
It's so descriptive.
the one that is.
That one behind.
Yeah.
Yes.
Yeah.
But we first go for the yellow one.
Alright, if it's.
She wants to zoom on the white one. We'll do afterwards.
I'm gonna call her.
Yeah.
Maybe with the limpets, you're thinking. I heard you say peripheral vent form, but
Looking at the peripheral fauna in Taiwan, they have like salad, sponges, shrimp, and stuff. So it's kind of like the concept of more idea. They don't even mention it as well. I was expecting to see them in this kind of community. So I just messaged him.
How far does the peripheral spread?
Not far. But we're not seeing any bacterial evidence.
and the temperature is staying constant. Well, no, it's not constant, but it's not.
Yeah, y del amarillo.
It's just that the limpets that I've seen different.
Yeah. Okay, también.
I haven't seen all that.
Okay. Vale. Pero esos los vamos a poner en quizzes. No de RNL.
Do we want to zoom? Before we click.
Yeah, do you want just to photo that?
Yeah.
Right
We
there.
probably.
Yeah.
We probably are at the maximum already.
Got it.
Yep.
Thank you.
Okay, thank you. Back to you guys.
Entonces, siempre queremos tomar una foto buena antes de mezclar y por eso es el código. Pero ahora que vamos a mezclar es una cabeza. Pero yo no tomo la muestra hasta que estemos confirmados del cobertura y ya después va hasta que se le pase. Para evitar confusión inicialmente. Y ya cuando esta muestra esté lista, ahí se envía una
Too much?
Try.
It's gonna be hard to cut.
Yeah, it's hard
Yeah.
to take like one branch.
One single branch?
No, that's not
If
easy.
you
I
cut,
think.
it'll be fine.
All right, we've been diving or streaming for about an hour. Just currently taking some samples here, but for folks just tuning in, this is live from the bottom of the ocean. You're watching Divestream 587, so that is the 587th dive of the remotely operated vehicle SuBastian. Currently we're streaming this to you from onboard research vessel Falcor 2, operated by Schmidt Ocean Institute. This dive is part of our ongoing expedition to better understand the vertical reefs of the Galapagos.
You don't want to make it too easy.
No. It's like when a ship.
And we are off of this pretty cool, just like flat-topped little rock out here, Roca Redonda. Currently it's a little too dark to see it out there. Instead our birds have shown up. As I understand it, that rock is home to many of these birds, so probably going to have a decent amount of them tonight. Started pretty deep with this one, so we're still over 900 meters down. We started a little ways down the slope from here even. And we are going to be making our way up into the 200s. So Nice little ascent there up this slope.
Come up, go down.
Oh, you see what's happening?
Yeah.
Can't change the current yet. You know, the best you can right there.
Here you can see the path we're looking to take. Got some waypoints up. We're going to get up to the top up here. Pretty shallow. I don't think we're getting all the way up to the top. We're going to get to about here, which is about 210 to 220 meters. 220 or so, I think, is what we're shooting for. And then we'll actually be coming back down a little bit if we have time at the end there. If we have time, definitely is a consideration for this dive. We've had no shortage of life so far. Lots of interesting things to stop and look at. We are doing video transects, but you know, this is different from what we've been seeing. So we are probably going to be stopping, doing some zooms here and there, taking samples. You might have heard Dan in earlier. He's not actually interested in the rocks right here, but we do want to try and collect some lava samples for him this dive. It's just that this talus field, right, it's all rock that has kind of slid down and accumulated. He wants rock that is more obviously in situ where it was, right? He wants to try and get some stuff that hasn't really just accumulated down here at the bottom of the slope.
Do you want it? Do we try again? Or
I mean, we're probably gonna have the same problem again, right?
We can move this
Try
up.
and move this up and see if I can get out of the current. That's about all I can do.
Are we positioned to take the white coral that is closer by?
So we're still in between our first two waypoints. And once we get to the next waypoint, 110, so we touch down at 19, then we're going to get to 110. Let me pull that back up, or at least put up your pip. So we got 19 on the far right there, then 110. We got to get up over this little hump, and then from 110 to 111. We actually see a sediment plane here, so that might be a little bit different again, right? Maybe a lot of polychaetes, maybe a lot of sea pens hanging out. We're gonna see. We're gonna see what's there, but there's certainly a lot of life here. A lot of currents, a lot of life, a lot of shrimp, and up top, a lot of birds, right? So it's a cool area for sure. Roca Redonda, the round rock. Like I said, we're about an hour in to our bottom time here. Total dive time, I don't know, it's got to span 12 to 18 hours or so. Need to have that ROV on deck really no later than about 0400 local time.
Yeah, I know. I almost thought it was a black coral on the side.
We are expecting to go through oxygen minimum zone, somewhere around 350 to 380 meters. So sometimes things are a little interesting around there. But the currents here are a little nuts. Let me actually put up the ADCP. So we talked about ADCP the other day, right? So that's the acoustic Doppler current profiler, right? So it's using sound and bouncing off of particles in the water, right? You can see all these little particles get carried by in the current, and that is enough by figuring out if the sound coming back, if the echo coming back is higher frequency or lower frequency than left, we can actually tell relative to us how fast and in what direction the current is moving at a given depth. So let me pull up, if I can, our ADCP. So you can see these crazy currents we got going on here. Alright, here we go. I'm just going to put them up in the picture-in-picture. You're really just looking at those arrows swinging around, just all over the place. Some of them are off the chart, literally off the chart.
On that yellow one? Are they different again?
So, it's just currents all over the place here.
Have we slurped some of those shrimps?
Yep. Sorry, Tyler, did any yellow thing come off?
No,
Inside?
no. It's super right out.
Okay. Alright, I haven't known that we have maybe a sample. We'll put it in and check and see.
All right, but yeah, you'll hear the scientists. They're all pretty excited to have touched down somewhere so abundant in life right now. So you'll be hearing lots of voices.
Those sponges I recognize from the Darwin and Hope dive.
Oh, yeah.
That they did. That's called Connerette.
The one with the...
Yeah, the one that had the corals on it, but that one is Connerette, and we have a nice sample of it in our collection. And I think that one got identified
Nice.
in one of the papers that got published. But yeah, I hadn't seen it. It's interesting that I see it here, but we didn't see it in the other bioregion.
So this new sample, is it okay to go in 3? Or?
Yeah,
Okay.
let's try to see. Earlier those sponges, I think, I said lollipop sponges. These are not, the lollipop sponge is fully branched, like lollipop. Your name is perfect.
Yeah, that's what I was
Yeah,
thinking.
they're
It's
totally
really
like,
But they look a little different to the tulips that I know
Oh, okay.
from the Northeast Atlantic.
yeah.
So can we first zoom on the branch that you plan to take? Yeah. Thank you.
Nice one, I got it.
Yep. Some of the pollets are closed already. Okay, thank you. Over to you.
Oh, you did not. You're the worst. You're the worst.
I thought you would take this branch,
That's
but
it. That's it.
whichever one is the
You're
easiest.
You're bad. I need to see that.
Yeah, because
That's
we
not
zoomed in
approved
on
for
this one. Yeah.
anything.
Oh,
other than delete it.
I'm gonna add,
Come on.
I'm gonna have permission from the park for that one.
Please don't do that. Oh, that's just.
Wait a control room sleeper here.
I closed my eyes for a second. You took a picture of me.
By the way, there's no film at all.
Yeah. There is, there's one.
Game on.
You snooze, you lose.
That's for sure. That's for sure. That's why I'm not sleeping anymore.
Sure thing. Don't worry, I've got one of Cloudy as well. Doing a little collection for the last day presentation.
Scrubs is easy though. It's easy stuff.
So I feel like here we're seeing a lot of species deeper.
Crisis origins.
Yeah. There's just the diversity is pretty high for usually I feel like this is around the 700s we start to see what we're seeing now. Including we just saw the bottle brush black hole. But for me it's like typical 300 and 600 anything. You saw one?
You saw that white? That white
the one
hole?
last night, yeah.
Tonight?
Yep.
Two together?
Already, yep. No, one I think.
Two together.
Oh yeah, the ones that we sampled yesterday.
Today.
Yes.
Half an hour ago.
Yeah. Yes, that was a different type of black hole.
Yes.
Yeah. That's it.
But we're seeing at least 3 black holes.
Yeah.
Come on.
Stuck on the side. Yeah,
no.
Oh.
There it goes.
Oh, the current is
It's a
really
floater.
Yeah, once you lift it out of there, it's gonna go away.
It goes, yeah.
Yep.
Okay, well maybe this is not a sampling dive.
Yeah.
Or
Is
it's a
there
slurp-
any
it's a
way to
slurping
get
dive.
it with the left manipulator
Or with
and
the slurp?
slurp it?
Can we slurp it?
The white ones seem to be high on the collection list.
And also try to push it. Alright.
Is it gone or did it go away?
So I've been coming in like this and letting go, but I can try to stuff it in head first.
Yeah, I
But
can
it
do
still
that.
might flow out because everything's getting pushed out. We can give it a shot.
Can you try it?
So you want to take another
So-
sample?
You can try another sample, the white one. If that doesn't work, we'll
Try that.
move on.
Try to get more
And there
items.
won't be much sampling
And
unless it's
also
with a
try-
slurp.
More like a bigger.
Yeah, just get the sample first.
Get more trunk.
Slightly
Go on the
bigger.
way, try
Yeah,
and
slightly
turn
bigger,
all
see
that
if it,
way.
it, it, it holds itself in more.
And then that way.
Yeah.
Yeah, free flying.
It's a great shot of that sapphire.
Is there one with an animal on it? Is that a crab? That might be hard to. Oh no, that's on the crassicles.
First choose your branch, and then before we take it, can you zoom into it?
Yeah.
Not many shrimp on top of here.
About this one right here.
The zoanthids.
Hmm?
Zoanthids.
Zoanthids, that's the thing.
That's what you're trying to think of.
Yes, and on the sponge.
Yeah.
Thank you.
That's what I said.
Oh my god.
That was
I'm sorry, but
That's alright. I should learn to
Turn it
talk
over,
properly.
turn your face down.
no.
No,
I was.
that
I think.
face.
What? Maybe when you said it, my brain was still trying to think, and I wasn't.
Just trying to get like, perpendicular.
Zoanthids. That's it. Yeah. Zoanthids.
Thank you.
Zoanthids. They are a colonial animal, right? So all of those there, even though they kind of look, they're all connected by some kind of swing.
Oh, sure.
Yep.
In the meantime, it's just going out of field of view. There was this reddish.
Coral?
Yeah.
Oh yeah, Paragorgia, right up here, right?
Yeah, sorry.
Yeah, yeah.
Yeah.
Or Swift, yeah, it could be a Swift, yeah.
We haven't got much Paragorgia.
Not in the beginning, the first, you weren't there for the first few days. Oh, I guess you were in the room probably staring at the screen. Yeah, maybe a bigger piece so that it has some traction inside of the tube. Yeah, so that it like.
You mean put your backside to the cart, yeah? Like the horses in the rain.
Like move away, like.
It's gonna block the current.
Yeah. I'm watching you, Tim. We're gonna get the angles this time. Have you seen these huge anemones? They're like 30 centimeters, they're like 2 feet wide.
I don't know.
I'll show you.
Purple.
Lavender. Brown lavender. That looked like it, didn't it? Yeah. Oof.
Yeah.
Was that one of our samples?
The sample is finished. We actually have 2 nets on the ROV.
Yeah, that might not be there when we come up.
Be careful. Somebody in the room with expectation of sleeping people.
Still awake, still awake.
great success.
Yes, that's true.
Slam the lid on that.
That's that one in quiver 3?
That's good.
Or quiver 2? 3?
But it was event 5, right?
Event 5, correct?
Yep.
Yeah.
What?
Okay. We continue now. See if we get out of 900 there.
Cool. Thank you very much.
No problem.
I guess we don't want to video transit.
We're not.
Still not,
We
more
made it about
corals,
30 meters.
more corals.
No,
So it was more of the same coral, it just keeps floating away.
Oh, actually, I did see that. Yeah. But the last grab went in, I was
Yeah.
hoping that if the current wasn't gonna take it and swamp that one out too.
Yeah, they turned
We had to
around
turn
to
around.
try and.
Oh, is that right?
Yeah.
You have to recover the sample in the lee of the vehicle.
Yeah.
That's
There you
right.
go.
Just
There
like
you
we're
go.
gonna have to recover the vehicle in the lee of the vessel.
Oh my gosh.
Yeah, that's some
Yeah, it's pretty
crazy
significant.
data up there. Yikes.
Is that yours?
The,
For recovery?
yeah.
Yeah. Oh, good
That's
luck.
what we look at. That will be fun.
Just have to turn all the engines on.
What's that?
You're
Just
flying
have to
against,
turn all the engines on.
Yeah.
against the current.
Are you bucking straight into the current or is it coming from the side?
Pretty much straight in right now, maybe a little bit
Yeah.
from the left.
It's like all it is.
Yeah. Well, once we get up over at the top, then it kind of levels out a bit, and there's a like a sediment field. It looks like
Yeah, we're crossing some sort of.
over here.
Yeah.
I mean, it's still pretty steep. It's like a little
Yeah.
ski hill.
I'm hoping that there is some outcrop at the top where all the stuff like that comes.
Sure.
It's not
Yeah.
completely covered in things.
I love it.
It's one of those sponges with not that many thorns. I said that there's one of those sponges,
yeah.
but not so many zones on top, so it's clear that it's a sponge.
But it seemed like those shrimp were only down in that one spot when you guys had.
No, the
them.
shrimp
Oh,
is
they're
still
still
here.
They're
here.
still there?
Yeah.
I don't have my glasses on. Oh, those are the red dots.
Yeah.
Yeah.
Oh, there you go.
Yeah, we were talking about this, right? They're like uniformly distributed.
Yeah.
It's great.
They have territorial limits of like 1 centimeter.
Yeah. Basically the extent of an antenna.
Exactly, right, there you go.
If I can touch you with the antenna,
Yes.
you're too close.
There you go.
Maybe they're just ticklish and they just don't want
That is, I like that explanation.
I have another one.
X-rated?
No. They're just kind of like Canadians or Anglo
Canadians, yes.
people who need space.
Oh, we need
They all
space.
need
Oh.
personal space.
Oh, I see.
Yeah.
There's a hatched out shrimp.
What? What's going on
Well,
with that?
that's a damn one.
Yeah. Oh, there's been so many of
Colonial
those.
siphonophore.
Yeah, a ton of them.
Oh.
They were one of the characteristic animals at the vent site at 86 West in the Galapagos Spreading Center when vents were first discovered. And they saw these things and they couldn't figure out what they were. And it turns out the closest relation is a Portuguese man of war.
A what?
Yeah.
Portuguese man of war.
Oh, right.
They're
Yeah.
a jellyfish, basically. Yeah. They are really super
That's
strange
close anemone.
with their
Oh,
tentacles
okay. Oh, I see.
stick them to the rocks. Yeah, there's a lot of shrimp.
Maybe not one of those enormous ones.
Yeah, no, there's an enormous one coming up right after.
This is definitely different than what we did.
Yeah.
Yeah.
I might zoom out a tiny bit.
Oh, real nice anemone here.
Just to let you know.
Is that one? Do you think that you got that in the miso camera in the front, or is it pretty much straight
Hoping
ahead?
so.
Wow, this one is huge. I've not seen these before. It's the first time I see
Yeah,
them.
Yeah,
I've
in
never
both
seen.
sides?
I've never seen one like that.
Trying to
No,
stay
and
low
none
enough
of the
to
guys
get
are
it.
Yeah.
None of your videos
Yes,
is
thank
there?
you.
No,
That's great.
it's like this big.
Yeah, it's big. I've seen ones like this that have very long tentacles, purple.
It's
Yeah.
like a pom-pom.
The Relicanthus, I think, is the long,
Yeah,
long
but
one.
this one is like
The
three
one in
times
the
the size
Antarctic.
of the pom-pom.
That's the one from the Antarctic waters.
We get, we were seeing up
Or
on
is
East
it-
Pacific
is it-
Rise and stuff. Yeah.
This is
Yeah.
sorry.
Super long, which was cool.
Yeah.
Definitely got a close up of it.
Oh, can we have a look at this? This is new here.
Sorry.
Which one?
Can we?
Sorry,
hold
go.
it from-
Where am I holding? What are we looking at?
I don't
A
know.
little
something
bit left.
slightly to
Can we
the left.
pan a little bit left? There's a weird chrysogorgid.
And I think of it.
Oh, here, down here, you see it?
I see an enormous crinoid.
Oh no! Wow, that is a big one. Look, that white thing that's floating here.
Yeah, I think I see what you mean. It's got a bit of a fan shape.
Yeah, can we just get a zoom on
Oh, that.
that? That is a different coral. I think.
Is that a coral at all?
I think it is. Yeah, I think it's a very white black coral. I know.
I'm sorry.
I don't know. It
What?
looks a bit glossy to me.
Can we get closer? Oh, you think it's
Oh no.
it's an albino black coral. I don't. Is it a massive hydroid?
Yeah, or a kind of spongy thing.
There you go, that's the one.
I'll move the camera a bit.
Yes, yeah, exactly.
Yeah, they're great.
Yeah, they're very cool. Very dramatic.
The Relicanthus is what we were talking about. The
Ah,
long,
the long
ones.
ones.
Yeah, they're fun.
Oh, I think it might be a massive hydroid. Yeah, no, I don't see colors.
No, I don't think it's. But given that everything is here bigger than it
Yeah.
is anywhere else.
I'm just gonna make a biological
That's
observation.
Oh no.
This is
Just drop it.
Do you have a snap?
Yep, I got one.
Yeah.
I don't know
Drop
what that
it.
is.
Let's everywhere. Thank you.
And while we are hovering here,
Oh,
I'll give you
nice.
a zoom of that large crinoid.
It's like Easter Bunny.
Also, is this
Oh,
is that
thank you.
someone for you?
Oh, we
Okay.
got it.
Oh, thank you.
Oh, that is big.
Oh wow. Oh,
That's a
really
pun.
skinny version for Stephen.
Oh, nice.
Brisingate or a crinoid?
I think it's
Crinoid.
Crinoid.
That is
Yeah.
But it has, yeah, you're right, it has all the markings of the procedures, but it's very skinny.
Yeah, no, it's a crinoid.
I'll stop this one.
Grand. Thank you. Shall I? I'm not entirely sure.
Would it be possible to display the forward-looking sonar?
Yeah.
Thank you.
Yeah.
They're just amazing massive. One of those anemones with the white tips.
But yeah, those bottle brush, these guys here,
More of those,
black rod. I'm surprised how deep they are here. That's crazy. Little bamboo column.
Right ahead.
Yeah, what is it? But do you guys know your sponges? Friday? I know everybody shakes their head.
Just stock sponge.
Glass. Just as good as we get.
What are these at?
That fly trap is giving itself all the possibilities there.
Verley, what depth did we start to dive at? Was it like 20 meters deep or 50
Like 940.
940?
Yeah.
Okay. So
Something around there.
down at 40 meters.
What would you describe this substrate as?
It's basalt talus. Good,
Talus?
good,
Yeah.
good angular pillow fragments, ranging anywhere from a few tens of centimeters to maybe half a meter for the larger ones. Probably with some manganese cover. I don't see any glassy parts to them, but I don't know.
I was looking for like rocky rubble fields.
Hard
But
substrate.
now I know even the possible composition. I love it.
So I've got them all there. How did they?
What got them all there?
Yeah, is it like a previous, like there were
it-
pillows, but then
There were pillows, and then there was probably faulting or mass wasting along this spine, and so they tumbled down. When they form, they have fractures in them because of the hot rock inside and the cold water outside. So they naturally fracture into these acorn shape or triangular sort of segments of pillows, and then they form talus that has that characteristic shape. I mean, if you look carefully at each of them, some of them look like they're full circles, but they all have these sort of radial fractures in them, and some of them are more triangular shaped depending on how they broke up. But so interestingly, there's not much sediment, so that means either the currents are just continuously active and not letting the sediment stay here. If we weren't in such a high current environment, you could say, well, it's an active talus, meaning it's recently fell down. In this case, I think the current is moving any any little bit of sediment off of these things and sending it somewhere else.
I also would say there isn't very much of a sediment source offshore Santa Cruz. You had a lot of
Yes.
much more sediment source.
Oh, absolutely, yeah. Well, both there and even on the seamount off San Cristobal, there was a lot of sediment. And I think that also speaks to the relative age, if you will, even though I think this seamount probably is older than Isabella as an island, right? So maybe 1 million years old or 1.5 million years old. I think it's likely that it predates Isabella, but hard to say. I mean, honestly, we've never gotten any good rocks from Roca Redonda.
That is
So.
a huge. Can we appreciate how big that top coral is?
The Muffinum, yeah.
Yeah, that is the biggest fat I've seen. Alright, we can continue.
Yeah.
Laura would be happy I know this.
Yeah.
That's a cool little sponge there.
Oh yeah, true.
How about the actual Roca Redonda? How, like, what is it?
The piece above water.
Yeah, why is that?
So
still
I
there?
think that is the volcanic plug,
The
right?
plug, so
Right, that is resistant but is getting eroded over time for sure. But I think that's the main plug, and I think what we're seeing in these spines are the rift zones that came off of it, that built what used to be the volcano. Now we're only seeing the sort of the skeleton of it, like, similar to what you see in the Western Pacific views and whatnot, but on a much smaller scale. Those are like 20 kilometers across, right? But it's the same sort of idea that you've got the sort of intrusive feeder complex is much more resistant to erosion than the pillows on the sides.
Very nice diversity.
Is that not the coral? No.
I think
no,
so.
No, it's a type of subcoral.
I think that was mother coral.
It sort of looked like it.
Unless it was a
There's
sponge
nothing
having a coral complex.
Yeah.
I guess we don't know anything about that sponge that looks like a coral. I mean, that's like, what's up
What
there?
is that, the other one?
We sampled
We
it on the
sampled it,
Atlantis.
but okay, yeah.
It's not a glass sponge, it's a demo sponge.
That's
That's something.
But you mean all of these yellow ones that we're seeing are not corals? They're sponges. The yellow
Oh,
things.
no. That
Oh,
that
this was
white-
something
that-
else.
yeah, it's one that we saw in a different dive.
That is interesting.
Yeah.
Can we ever look at this golden coral here?
The
Sorry.
Candlestick one, yeah.
Yeah, that's looking a bit different.
We've come across a couple of, or I've seen it 2 or 3 times already on this.
But looking that healthy and
One looked like that.
I think it's in a comfort board too.
And it's bushier than the other
Yeah.
one that we've been calling Mercedes.
What are we thinking? I don't know. What's been sampled?
You're
or not.
happy.
Do you think it's gonna be the same flying out of the
Can I zoom?
It's like the current's moving the other direction over here actually. Before it's gonna go from left to right, unless it's moving right to the top.
It's a
Do
black
you
coral as
need to
far as
keep
I can
these
see.
all separate from each other? You can't put them in a bio box because it'd be easier to trap them in the bio box.
But I mean, if we open it again, it'll fly out of
Maybe.
the bio box,
Maybe.
maybe. I don't know enough what that's been sampled, so have that one.
It's quite difficult to get a
I
fixed
mean,
zoom on it.
to me it
Keeps
looks
moving.
different to what we have.
Let's give it a try. We
Can we try to
clip it one at the base and to be very long, we might be able to put it in.
Very nice polyps.
Yeah.
That looks great.
Octocoral.
Beautiful shot. Nice.
Do we sample it?
I'm down if we have time, but I don't want.
Give it a try if you want.
Okay.
Stuff.
Event 6.
It's easier to do on the rail, just sticks up. Still good.
Number 4? Quiver number 4.
Is there something in there? No?
No, I don't think so.
No, it's number 3. It seems like you should be on at least number 4 by now.
Oh, you would be bored if you don't get any samples to do.
Why is it that? Is iron in there?
Fresh
water.
water.
Oh, okay.
Yeah,
Oh,
if
of
it
a very
rises
different
up out of it,
temperature.
it's our fresh water. If it stays down in there, that's the RNA later. Yeah. It's cool to see the differences.
Once you've
Want
chosen,
to?
yeah. If you don't mind.
Yeah.
Cool.
Thank you.
Yep.
Open up.
Is that the close up of the sample?
Yeah, that was part of it.
Yeah.
Merlin, can you get even closer at the polyps when they're done grabbing it?
Okay.
Like a really close up of the polyps? This one has really large polyps, so I think we can get a really good one.
But I suspect by the time it's grabbed, the polyps will be closed. We had a really good one
Okay.
before.
Okay.
Yeah.
Nice. Just get a dunk.
Did you see that the shrimp came and checked out the sampling action? Oh look, there's the sponge with the coral complex up there. I'll show you. There's a sponge that looks like a coral complex. dabbled in recycling. But that's fun. Oh, 06?
Oh, 6. Very slowly.
Still a bit stuck.
There we go.
Yay! Oh,
Very nice.
That is 0.2867 million. Longitude is minus 91.649666 at 85.9 meters. This thing here is like a tube worm extravaganza.
Yeah. Woohoo.
You see? See you guys? Yeah, someone in the chat has confirmed with me that those curly sponges are Faraday.
Ah, okay. The ones that get
Yeah, I think it's Conorette, that's the genus, but it's definitely a Faraday.
Mm-hmm. There's a bamboo. There's a bamboo coral.
Yes, it's a big bamboo coral. Oh, qué bonito. You can probably see the bamboo
Yeah. That's
body.
can
And this
we
is a beautiful,
zoom
what
in?
I call a mixed garden of
Yeah, go for it.
glass sponges and soft colors. Bonito.
Thank you.
Oh, nice one.
Nice polyps as well, right?
Yeah.
Just gonna look at the base so people can see the bamboo because it has
Gotta go
these
low. You gotta go
black
low.
knots. Yeah.
Oh yeah, oh I see it there, yeah.
Yeah.
Nice.
So that's how I saw it was a bamboo coral.
Yeah. Looks pretty old.
Yeah. Thank you.
Right, swaddling would love a piece of that.
Yep. Good. So much cool stuff in general.
Yeah.
Sponges as well.
The shrimp have been going on for a good 100-meter depth.
Yeah.
Yeah.
Don't seem to change in density either.
Not really, no.
Just how far like the
There seems to be a tiny bit of sediment now. Just select this one. Just to want to tad. Yeah. What's that spindly thing? Spindly coral.
I think that is kind of like we sampled something like that earlier, but the white one that we sampled, but I think this is a more haggard version.
Yeah.
It might be something else. So many fish are gorgeous. Quite simple. And needless to say, a lot of anemones.
Yeah. I was thinking this like lots of fly traps.
And those sponge tulips that I'm a fan.
Something large. Might be sponge.
That is I think that's a bamboo coral as well.
Yeah, that's a sponge. The one that's just passing.
Yeah, yeah.
Yeah.
The black coral. Can we get a zoom of this bushy bamboo coral? Just the zoom.
This one?
Yeah, that's,
Yeah.
that's a bushy bamboo coral.
Can we see the ROV on the Flighter mouse screen? Is that
Yeah, let's see what we got.
I'm curious where we are on the
We're not
ascent.
anywhere we think we are. I think we're tucked in. I think we're right
We might be
here.
at the. Okay, we're under.
Yeah, I mean, I can.
Underway.
s-
No,
I
that's
can-
cool. I was just kind of curious.
Oh, where'd my? Oh, there it is. Yeah, you can just zoom straight. Yep,
Oh, yeah,
that's
right,
us.
Okay, I got a snapshot. Oh, you're getting closer. Nice.
I
So
really
we're
has the
like
polyps out, so
there.
Halfway. Hard to see the top.
Yeah. Yeah,
very nice.
we're gonna get there.
It's different to the other bamboo corals. It's a lot more bushier, right?
Yeah, and much thinner branches, right?
Yeah.
Thank you.
See, those are the corals I expect to see more around this depth.
Oh, that's a cool rock there.
Yep.
Actually, 100% is that. It's great. That's a good one.
Sorry?
Which one?
Either it's a sponge with some runteds, or it's a bamboo stalk.
Look at this dead bamboo coral.
Yeah.
Shall I zoom into it?
Yeah. Honestly, I feel like Laura would love one of these.
Oh, nice little star on the right there too.
Yeah, nice push. Sorry?
Just behind there, there's a bit.
Yeah. Although the one behind seems to be, zoom in view, seems to be, could be a dead sponge.
Weird.
But-
Like part of it is still alive.
Is it still,
cup coral there.
so there's desmophilins on it, there's
Yeah.
a stillasterate on it, and then there's polyps. Are these polyps native or is it something else encrusting?
I don't know. I mean, it looks like bamboo. It could be bamboo coral polyps. It's not. There's one. There's one that is right next to it. On this bamboo.
But there seems to be a sponge kind of tucking in between there as well.
Yeah, right?
Yeah.
Weird.
The whole ecosystem on its own.
I don't know.
Very cool.
Yeah.
Thank you.
Oh look, that's the piece of the bamboo coral that fell off.
Yeah, I wasn't sure. To me, it looked like a sponge, but maybe it is the bamboo
Yeah.
that's encrusted by a sponge.
Yeah.
There's more of this very fine, thin bamboo as well.
So this looks a little bit like what it was going up when the ocean exploration trust went out to the Darwin and ROV sea.
Okay, yeah.
The shallowest one. It's kind of like this with these lots of these white kind of like curly sponges and
Uh-huh.
lots of yellow acanthophores.
Okay, nice. Yeah, there's definitely a few different species of yellow. Cool. What was that?
Yeah.
Yeah, yeah.
Yeah, one's a swiftly and the other one's a acanthophore.
The rocks seem a little smaller now. Oh, black coral.
Yeah,
Nice
black
feather.
coral.
Yeah.
I kind of don't want to cut, and we're entering, so we're gonna enter the zone where we're gonna be sampling for cover number 12, so I'm holding back on this.
I will sample
Yeah, yeah, yeah.
extra
So we can
stuff while
actually
we
get
sample.
somewhere.
Well, we have another 100 meters to go. More than 100 meters to go.
Are you doing some BBC today? Tomorrow.
Wow, some very cool stuff there.
Let us know.
I've seen any of this stuff like this.
Do you know the depth of this thing? No, I didn't check. It was definitely the biggest knob there, like it was different from
Oh, you're trying to
Like the top of the knob.
top of the knob, yeah.
837, 35, somewhere in there.
Okay, so we're pretty close. 8:35.
Oh, that's another 50
Oh, there's
meters
a
up.
white fish in the sponge.
Yeah.
Oh, look at him.
That must be on purpose, right? Like he knows he can be seen less in there. Not stupid.
All right, been about 2 hours on bottom. For folks who maybe tuned in just in the last hour here, this is dive 587. Currently working our way up this slope. Pretty early in the dive still. We started down at like 945, 945 meters down. We're at about 865 now. We are hoping to get up to about 220 meters shallow. So we've got a ways to go. This particular dive is on this cool little rock feature. You can actually see a lot of this here. Someone asked if we'd already collected rock. We have not, and that is because this talus field is rock that's fallen from above. We want to try and get rock that's still kind of on site. So we're hoping to find that a little further up, maybe where all this rock is coming from. This features actually Roca Redonda, so it means round rock, and it is quite cool. Wikipedia's actually got the wrong photo up for it. It has another Redonda. I don't know why. The article, if you actually check out the articles, because we were all like, oh, let's look up. The article says not to be mistaken for these other islands with similar names, and then the photo on that page is of one of those other islands. But this is a little flat top rock that just kind of juts straight out. I actually showed it way back at the beginning of the dive, just about 2 hours ago. Briefly showed it while it was still light out and we could see it on the camera.
You're working on the yacht? Yep.
We are just working our way up, doing these transects. We're actually headed up. We're hoping to reach the top of a little knob sticking out the side here at about 835 meters or so. Then we're going to dip back down. We're going to cross over what looks like maybe a sediment plain area. Kind of looks like a little avalanche chute or something like that. And then we're going to continue our way up until we're about like 220 meters shallow. And if we have some time, we're going to then dip back down just a little bit. Lots of life here. The shrimp are getting less numerous for sure at this point, but when we started out, there were just shrimp everywhere. Pretty cool. You can still see them. They're still around, but they are getting less dense, I would say, at this point.
Yeah, it's cool. It looks almost like a whelk's egg case, but
Yeah, right.
But it has those sort of circular things.
Yeah.
But
I have no idea what that
I
is.
think that is what it is. I don't know what it is.
Picture.
It's quite.
Yeah.
Did you think that they were eggs attached to the rock?
I thought something, yeah. Maybe, I mean, probably not a sponge.
Unidentified white object.
I don't know.
Oh, look, is that more?
Can you just zoom in on
Yeah, there's more at the bottom.
Because the question is, are they individual or is it all one thing?
There's more of them.
They definitely lined
Yeah, there's a bunch right there too.
That you mean the thing down on the bottom of the screen?
I would
Yeah,
Yeah,
there's
that
more
thing.
there as well. In the meantime, very nice color change on your rock.
Yes, except it's not a good color change. It's all iron oxidized.
It's still very pretty.
Yes, it is pretty. Yes, I'll give you that.
Yeah. Alright, I'll zoom out.
Yeah, we
There
don't
are
have
some-
to linger on this.
There are some
We need to
pieces here
make
that
progress.
look like they're sampled.
Yeah.
And so Dan, you're looking for stuff that's still basically where
Yes, that's
it
in
came
place,
from.
yeah.
Where it starts.
Exactly.
Yeah,
Before
yeah.
it falls off into the rest of the pile.
The source.
I feel like the shrimp are slowly getting a bit
Can we,
sparser.
They are. I was just noting
just
that,
hang there for
yeah.
a second?
Yeah, I'm just
They're still around. They're still
Yeah,
there,
yeah.
but much less of them
So
now.
I'm
Yeah, you
Is that
you
okay?
point what
okay.
you would like.
So this stuff here looks like it's gravel.
We might have found a rock for Dan here. We're gonna see. We're gonna see what we can do.
Pieces that look like they.
So yeah, that's about where we are now. It's just we are actually still in between our first waypoint and our second waypoint. So a slow start, but that's because it's been an amazing start. Lots of life, just an abundance here, which is very cool. For folks joining us, welcome. You know, this is a good one. We're going to be going through the night. We're looking at latest, we want to be back up on deck at about 0400 in the morning. So we are looking for that.
It's more loose than I thought.
Yeah, you made some samples.
Let Dan grab
If this is your first time, welcome again. This is actually live footage. So this is live from a remotely operated vehicle. Think like a robotic submersible, right? No one's riding in the submersible. We are actually watching the footage along with you on a ship on the surface, the research vessel Falkor 2. And our pilots are sitting in here on Falkor 2. They watch the same cameras that you see through these streams, and they use those to pilot the ROV, to actually fly it around almost a kilometer down.
Yeah, it's very diverse.
So we're going to take a look at this rock here. It seemed pretty crumbly, so they're going to try and grab a little sample of it. But yeah, first time joining us, and you would like to see more of this, you'd like to learn more about the deep sea as it is being understood, as scientists are exploring it, go ahead and subscribe to us on YouTube, or maybe you're catching us on Facebook. You can follow us over there as well. But if you want to learn more about this specific cruise right now, I'm going to post a link up for you. And you can find information about all of our cruises over at schmidtocean.org. But I'll post the link to this particular cruise, which is the vertical reefs of the Galapagos. So we are looking for basically corals growing on these vertical faces or very steep slopes. Difficult to map because most mapping is done in a top-down manner with sonar. And so we actually have done some mapping dives with this expedition as well, where we've put mapping equipment on ROV SuBastian facing kind of forward, not just down, and then we've flown down next to these vertical faces and scanned them. So some pretty cool tech being used here.
That's true, because today when I was doing this talk, I did once, I can repeat, but nevertheless, going on all the steps,
If you are watching along live on YouTube or Facebook, you can always type questions in the chats there. We do keep those, or keep an eye on those, and try to get answers to your questions when we can. Dan has hopped on over into the science seat, just to kind of point out to the ROV pilots here the samples that he would like. Yeah, we were discussing these white little structures adhered to the rocks. Folks here weren't sure if they were eggs or some other type of organism. Hopefully we can get some really nice zooms on them. In a little bit, I'll be hopping off, but the stream's going to keep going. I think Juliana will probably jump in here at some point. But you might also have Rich through the night, or you might get Josh on in the morning. So we are planning to stream the dive. Little isopod cruising around there if you got eyes for it. Always fun to see them swim.
Yes, event 7.
You see this way.
So the rock here is pretty flaky. It seems like it's really quite crumbly.
It is a bit manganese covered.
Can you tell us the name of the box?
Rock box 1.
Dropbox one.
And if you don't mind, Tyler, to try to go back and just get another.
Dan, tell me the number of the rock again.
RB1, Lockbox 1.
No, the number of your rock sample.
28.
28.
Okay, so Dan, we were kind of picking right around here. That's where we were picking.
Oh, this
It's the age-old biology versus geology.
Back in with this.
Yeah, quite a few rock pens in this area as well. Keep an eye out for those. Very Dr. Seuss looking.
Yes. And the rest of the cliff too. Okay, that can go on the same box, please, and it's the same sample.
Okay.
2 pieces.
So now you just write
Yes.
times 2 more.
That's where we go to get this stuff. And just on
Let me know when you're ready.
Should be good there.
A little boat up on it.
Yeah.
Shoveled her up.
All
Right after the rock sample.
2, 8, 6, 7, 4, 4. And then longitude is minus 9, 1.64, minus 3, 2, 8. The depth is 8, 5, 9, 7. Back for the photo album.
Seriously?
Can you take a picture of that, please? Oh,
Yeah.
Sorry about that.
it's all right. No, that's fine. Okay, we're good. That's a great rock. Thank you very much. Appreciate it.
Anything else from here?
No, not for rocks. I don't know what else they want to do, but we'll leave that.
There was a request for a short
Okay.
moment, but Ollie walked out.
Oh, Ollie wanted to do some filming.
All right, so we are going to just take a quick little intermission in this spot here, and we will be back in a bit.
Oh, here it is. Sorry, I got it.
I wasn't on the lead.
Oh, that's okay.